All right, let's dive right into this explainer. We're going to be tracing an absolutely epic 3.5 billion year old story today. We're looking at exactly how life evolved from a violent, lifeless chemical soup into the highly sophisticated protein factories operating inside your body right now. It's a total masterclass in molecular engineering, and to really understand how you function today, we have to look back at the very first blueprints.
Here's our roadmap for today. We'll start with the spark of life, move on to the RNA world, and then look at reading the DNA blueprint. From there, we'll hit the protein assembly line, check out folding and upgrading, and finally, look at what happens when the blueprint glitches.
Okay, part 1, the spark of life, or Earth's primordial soup.
To really wrap our heads around our cellular machinery, we've got to travel way back in time. I want you to picture early Earth. It was violent, chaotic, torrential rains, nonstop volcanic eruptions, and zero ozone layer to block all that harsh radiation. So it brings up this ultimate mind-bending question. How on earth did simple, random chemical reactions in that kind of extreme environment organize themselves into a complex, self-replicating organism? I mean, how do you actually get from raw elements to life?
Well, the famous Miller-Urey experiment brilliantly illustrates the answer. It's actually kind of crazy. If you take the simple gases that were just floating around on early Earth, stuff like methane, ammonia, and hydrogen, mix them up with water, and blast them with electricity to simulate early lightning, absolute magic happens. You spontaneously generate the amino acids, the sugars, and the nucleotides that are literally the basic requirements for life. The building blocks were just sitting right there, waiting in the soup.
Moving on to Section 2, the RNA world, the ultimate multitasker. Now, having raw materials isn't enough, right? You need a system. And the RNA world hypothesis gives us that missing link. Think of RNA as the original overworked startup founder before DNA even existed. It is the ultimate multitasker. On the one hand, its sequence of nucleotides acts as a genotype, carrying the actual genetic code. But on the other hand, it can physically fold itself into these complex 3D shapes to act as a phenotype, a functional catalyst or a ribozyme that actually drives biochemical reactions. So it was the blueprint and the builder all rolled into one. But there was a catch. If RNA synthesized a really useful protein out in the open ocean, it would just float away and end up helping the competition. For a true cell to form, they needed containment. It's super cool how this happened. Amphipathic lipid molecules, which basically just have water-loving heads and water-repelling tails, spontaneously clumped together to form a secure, enclosed bubble. Once that happened, the RNA could keep all those beneficial proteins it was making entirely to itself. That was a massive, game-changing evolutionary advantage.
Okay, Section 3, reading the DNA blueprint, the master vault. As evolution marched on, this primitive factory got a major upgrade: DNA. DNA is this highly stable, double-stranded master vault of genetic information. But here's the thing, you can't just build proteins directly from the master vault. You need a working copy, and that process is called transcription. An enzyme called RNA polymerase literally unzips a section of DNA to build a complementary strand of pre-messenger RNA, and it does this at a blistering pace. We're talking 20 nucleotides per second. It whips up this temporary, highly accurate working copy.
But before that temporary pre-mRNA copy is allowed to leave the nucleus, it's got to pass through the factory's quality control. The cell pops a 5' cap onto the front to protect it, and then slaps a long poly A tail onto the back to keep it stable. Finally, this massive complex, called a spliceosome, comes in and literally snips out all the non-coding filler regions, which we call introns. Only after all that heavy processing do we finally have a mature, ready-to-use mRNA blueprint.
Which brings us to Section 4, the protein assembly line. This is the modern factory.
So once that perfectly processed mature mRNA gets exported out into the cytosol, it hits the actual protein assembly line. This is translation. It's an incredibly tightly choreographed process that relies on 3 main players. First, you've got the mRNA blueprint itself, carrying instructions in 3-letter codes called codons. Second, the ribosome, which is the actual manufacturing machine. And third, transfer RNA, or tRNA. These brilliant little adapters read those codons and deliver the exact specific amino acids needed to build a growing chain, usually starting with methionine, by the way. And the sheer speed of this assembly line is absolutely staggering. The ribosome zips along that mRNA, chemically bonding amino acids together at a rate of up to 15 amino acids every single second. Plus, you can have multiple ribosomes reading the exact same mRNA strand at the exact same time. They are just churning out identical proteins in an absolute explosion of microscopic industrial efficiency. Next up, Section 5, Folding and Upgrading, the Packaging and Shipping Department.
Now, just producing a raw, straight chain of amino acids, that's actually kind of useless on its own. It still has to go through the factory's packaging department. First, the chain has to fold itself into complex 3D structures. Then, it goes through what we call post-translational modifications. So proteases might snip the chain to activate it, or the cell might tack on methyl groups, attach complex sugars, or form these super strong covalent bonds, like disulfide bridges, just to lock its 3D shape firmly into place. These modifications are totally crucial. They dictate exactly what the protein is going to do and where it needs to be shipped.
Finally, Section 6, When the Blueprint Glitches, the Real-World Stakes.
We really need to anchor all this microscopic stuff to human reality, because when this multi-billion-year-old assembly line makes a mistake, even the tiniest, almost invisible mistake, the stakes are literally life and death. Let's look at sickle cell anemia. Out of the billions of letters in your DNA master vault, a single typo swaps out just one amino acid, changing glutamic acid to valine in the hemoglobin protein. That tiny little glitch causes the entire protein to misfold. The red blood cells become rigid, they take on this sickle shape, and they end up blocking blood flow. One wrong letter, massive systemic consequences.
Similarly, a glitch in this assembly line is at the very core of cancer. If a mutation happens to hit the RAS signaling protein, it can literally get stuck in the on position, and that tells the cell to just divide endlessly. Or, if the p53 regulator gene gets broken, the cell completely loses its ability to initiate apoptosis, which is basically its self-destruct sequence when it's damaged. Without those functional protein regulators doing their jobs, the factory just runs completely out of control, eventually secreting enzymes to break through surrounding tissues and spread, or metastasize, throughout the body.
It is honestly mind-blowing to think that the legacy of a 3.5 billion-year-old chaotic chemical soup has evolved into this flawlessly regulated, lightning-fast factory operating inside your cells right now. Every single second, trillions of these molecular machines are transcribing, translating, and folding the exact proteins that are keeping you alive. So I'm going to leave you with this final thought to ponder today. With trillions of machines working away, what is your blueprint building today?